Glycolysis harvests chemical energy by oxidizing glucose to pyruvate. ATP is used to prime a glucose molecule. These three carbon intermediates are oxidized to two molecules of pyruvate, yielding a net of 2 ATP and 2 NADH. ATP is formed by substrate level phosphorylation, in which a phosphate group is transferred from an organic molecule to ADP. The second step in glycolysis. The second step in glycolysis is the isomerization of glucose 6-phosphate to fructose 6-phosphate. The carbonyl oxygen of glucose 6-phosphate is shifted from C1 to C2. This results in isomerization of an aldose, that is glucose 6-phosphate to a ketose, fructose 6-phosphate. Enzyme which catalyzes this reaction is called either phosphohexoisomerase or phosphoglucoisomerase or glucose phosphate isomerase. The enzyme facilitates conversion of pyranose ring of glucose to furanose ring of fructose. Isomerization has two relevances. First, in the subsequent step of glycolysis, when phosphorylation occurs at C1, the hemiacetal group OH of glucose 6-phosphate would be more difficult to phosphorylate than a simple primary hydroxyl OH. Second, for carbon-carbon CC bond cleavage, which occurs in the next step, isomerization to fructose with carbonyl group coming to C2 activates C3, making cleavage of the CC bond easy. In humans, this enzyme requires magnesium ion for the activity, and it is highly specific for glucose 6-phosphate. The delta G naught prime value is 1.67 kilojoules per mole. This small value means that the reaction operates near equilibrium in the cell and is easily reversible. In the previous reaction, carbonyl group of glucose 6-phosphate moves from C1 to C2 portion and the hydroxyl group at C1 becomes available. Phosphorylation of this group occurs, which is catalyzed by the enzyme phosphofructokinase PFK. Similar to reaction 1, phosphoryl group is provided by ATP and requires magnesium ion. Phosphorylation of fructose 6-phosphate is endergonic and is another priming reaction. Reaction catalyzed by ATP dependent PFK is essentially irreversible. At pH 7 and 37 degrees centigrade, reaction catalyzed by phosphofructokinase lies mostly toward right, similar to the reaction catalyzed by the enzyme hexokinase, and is irreversible. Conversion of fructose 6-phosphate to fructose 1,6-bisphosphate catalyzed by phosphofructokinase is a committed step for glucose to be used for energy releasing process rather than converting it to another sugar or just storing it. Thus, reaction catalyzed by PFK is an important regulatory step in glycolysis. ATP acts both as substrate as well as allosteric inhibitor of the enzyme. Therefore, phosphofructokinase has two distinct binding sites for ATP, a high affinity substrate site and a low affinity regulatory site. In the presence of high ATP concentration, phosphofructokinase subunits behave cooperatively, that is, the activity of one subunit influences the activity of all other subunits. When enzyme activity is plotted against fructose 6-phosphate, a sigmoidal curve is obtained, which is typical of an allosterically regulated enzyme. Inhibition is reversed by AMP. Rise in the levels of AMP within the cell is coupled with decrease in ATP, and it is the ratio of ATP-AMP which regulates the activity of phosphofructokinase, that is PFK. Thus, the activity of phosphofructokinase depends on the levels of both ATP and AMP and is a function of cellular energy status. Glycolytic rate is decreased when ATP is in excess and increases when more ATP is required by the cell. Pyrophosphate dependent PFK. In plants, an additional cytosolic enzyme PPI dependent phosphofructokinase is present which phosphorylates fructose 6-phosphate using pyrophosphate in a reversible reaction. The PPI dependent PFK is present in quite an appreciable amount in the cytosol of plant cells. But ATP dependent PFK can take over the function of PPPFK1 in case of its absence or is present with reduced activity. This shows that quite a flexible system present in plants to provide more alternative reactions. Other molecules which are potent allosteric activators of PFK are fructose 2,6-bisphosphate and ribulose 5-phosphate. Besides, in plants, the activity of PFK is also inhibited by PEP. Thus, there are multiple layers of regulation for the activity of PFK, which indicates reaction catalyzed by this enzyme to be the primary regulatory steps of glycolysis. Now let's have a look on some MCQs on this topic. Question 1. Phosphofructokinase is characterized by which of the following statements? It is a major regulatory enzyme in glycolysis. ATP is a substrate of the enzyme. ATP is a negative modulator of the enzyme. Citrate is a positive modulator of the enzyme. Answer C. P, Q and R. Question 2. Which of the following enzymes in glycolysis catalyzes a reaction that is essentially non-reversible? Enolase, triose phosphate isomerase, phosphofructokinase, phosphohexose isomerase. Answer C. Phosphofructokinase. Question 3. In glycolytic pathway, acetyl coenzyme A acts as allosteric inhibitor of pyruvate kinase. Enolase activity is inhibited by fluoride. Citrate acts as allosteric inhibitor of phosphofructokinase. Hexokinase requires divalent metal ion such as magnesium or manganese for activity. Answer B. PQRS. The glycolytic pathway requires which of the following as allosteric regulatory enzymes? Glucokinase, phosphofructokinase and pyruvate kinase. Hexokinase, phosphofructokinase and pyruvate kinase. Hexokinase, glyceraldehyde 3-phosphate dehydrogenase and enolase. Phosphofructokinase, enolase and pyruvate kinase. Answer B. Hexokinase, phosphofructokinase and pyruvate kinase.